Good afternoon, everyone. We'll go ahead and start the meeting here in just 1 minute. Okay, so I know people are still joining, but welcome everyone to Farm to Fork quarterly virtual meeting. We are excited to bring this quarterly meeting back and look forward to uh, the kind of the evolution of where we go from here. So without further ado, I just want to provide a brief disclaimer. So although the content of the Farm to Fork quarterly meeting is directed towards veterinarians, epidemiologists, public health professionals, and federal, state, and local positions, as well as industry members, USDA does not have control over who participates on this call. Therefore, please exercise discretion on sensitive content and material, as confidentiality cannot be guaranteed. And this virtual meeting will be recorded. If you have any objections to this, you can disconnect. So as kind of one aspect that we'd like to kind of introduce and bring to these Farm to Fork quarterly virtual meetings are any updates that are happening within the pre-harvest food safety space. So we wanted to let everyone know that our colleagues at FSIS is announcing its proposed determination that raw chicken carcasses, chicken parts, comminuted chicken, and comminuted turkey products contaminated with certain salmonella levels and serotypes are adulterated within the meaning of Poultry Product Inspection Act. So FSIS has proposed this new document and invites interested persons to submit comments to the document. Comments on this proposal must be received on or before November 7th, 2024. And I will provide a link with further details on that salmonella framework for raw poultry products that links to food safety inspection services and where you can find more information on comments. And then there'll be a presentation today on commensals versus controls and salmonella in boilers. This presentation will be given by Dr. Ryan Arsenault. Dr. Ryan Arsenault received his PhD in biochemistry from the University of Saskatchewan, conducting a doctoral research on the Vaccine and Infectious Disease Organization and studying Kinomyces, prions, and Johne's disease. He did his postdoctoral training with the Agricultural Research Service studying foodborne diseases of poultry and cattle. Dr. Arsenault was a professor at the University of Delaware for 9 years in the Department of Animal and Food Sciences. His research in Delaware Kinomyces and the gut health of poultry and included topics of immunometabolism, post-pathogen interactions, feed additives, and antibiotic alternatives. Also at Delaware, he helped create their One Health certificate program. In 2024, Dr. Arsenault took up his new position as research lead for the Food and Feed Safety Research Unit of the USDA Agricultural Research Service in College Station, Texas. In this role, he leads a dedicated team of multidisciplinary scientists and technicians researching means of reducing pathogens of foodborne concern in live food animals with the goal of improving food safety and eliminating human infections. Dr. Arsenault will give a presentation on the commensals versus control of salmonella in boilers. If you have any questions, please provide them in the Q&A function of the chat. And without further ado, I'll pass it to Dr. Arsenault.
Perfect. Thank you for the introduction. Hopefully you can see my slides there. So I'm very happy to be invited to present at this meeting. Very happy to present some of the work we've been doing on pre-harvest food safety, specifically with broiler chickens and specifically with salmonella. So certainly this group doesn't need any introduction to One Health, but I do like this definition of One Health as an emerging concept that aims to bring together human, animal, and environmental health. And One Health, I think what's interesting about it, in addition, is that it really encompasses a lot of the hot topics that we're dealing with as a society, as a planet. I think this figure illustrates that well, from deforestation, biodiversity loss, air and water pollution, climate change, social determinants of health, diet, food security, antibiotics, antibiotic resistance, vector-borne diseases. Pretty much everything we're dealing with as a planet is sort of encompassed in One Health. And really the power of One Health, which I don't have to, again, explain to this group, is that we're bringing these interdisciplinary groups together, or at least that's the goal. The sort of the cliche of knocking down these disciplinary silos and getting people to work together. I like this quote that it's specifically about universities, but I think it applies to scientific disciplines in general. In the absence of an integrated curriculum in which each discipline informs the other, each part making sense in light of the whole, the modern academy has literally disintegrated itself into a plethora of fragmented particles, none of which is in communication with the other parts. And I think One Health is the vehicle to get that communication to work, as long as we're doing it in that spirit of equality and of bringing in all comers to have an equal say. I like to paraphrase G.K. Chesterton, and when he said, I would apply it to One Health. It's not been tried and found wanting. It's been found difficult and never tried. Where we actually consider that triad of One Health as equal. And one of the I could give numerous examples, but even when you're talking about One Health, and this is from a paper, and this is just one example I picked. There's many others that I could have chosen, where you're talking about foodborne illness, you're talking about it in a One Health perspective, and yet all arrows, all signs seem to always end up pointing to humans. And we don't have as much of that bi-directional, or if it is, it's a dotted line and a solid line, or if it's a solid line and no reverse, and somehow that we would never infect a food animal with pathogen that came from humans, for example. We would never contaminate food animals with salmonella, for example. That's an example of not really taking an equal perspective when we're talking about some of these big problems in One Health. And today I want to talk about salmonella specifically, salmonella in broilers, a major foodborne illness concern. And food safety really is integral to One Health. I think of agriculture and I think of food safety as really the epitome of One Health, where they really sit in the center of One Health. There's nothing you could think of that impacts the triad of the environment, of humans, and of animals more than agriculture and the production of food. We use the environment to grow the food. We use animals as a food source. We're feeding the human population. It really, there's nothing that's a bigger One Health topic than agriculture. So specifically in the chicken, salmonella, it's everywhere in the chicken's environment. It can contaminate feed. It's certainly present in the housing environment. It could be introduced through the environment of soil, flora, and fauna. It can colonize the chick right from hatch or really even pre-hatch. If you've got a salmonella infection in your laying hens, that those bugs can invade the reproductive tract and actually inoculate an egg pre-hatch. But certainly post-hatch, you've got all of these elements coming into play to potentially contaminate the bird, and that can persist as the chicken grows through grow out, and that persistence is really the theme of our research and what I'll be talking about today. And the oral inoculation of these chickens, the salmonella can reside anywhere along the gastrointestinal tract. One of the major sites that we look at is the ceca, these two sort of blind sacs here off the large intestine. Salmonella really like to live and persist here, but they can persist throughout the gastrointestinal tract. And what we've studied when it comes to salmonella and chickens is that they mount this very interesting immune response. There's this host pathogen interaction that's it's not totally unique to the animal kingdom, but it is quite unique in regards to the animals that we generally talk about, that they tolerate the salmonella. So we've measured this in a number of different ways. In this case, we looked at CD4 positive, CD25 positive cells and their expression when we inoculate chickens with salmonella. And what these cells are is they're T regulatory cells. They're T cells, part of the immune system, but they're regulatory in that they return the system to homeostasis. They downregulate active adaptive immune responses. And when we inoculate young chicks with salmonella, at day 4 in the infected birds, and through day 7, and then a big jump at 10 and 14, these cells get induced. So there's this big jump in these regulatory homeostatic cell type. They also produce this cytokine called IL-10, which is again a homeostatic sort of dampening cytokine, immune protein that's produced by these cells, and it'll turn down an active immune response. And we see this run up in the production of IL-10 from day 4 to 14 post inoculation with salmonella in these birds. So you're really seeing this induction of a tolerance to salmonella and a dampening of the immune response to them. We've measured this in other ways. This looks at two cytokines, IL-6 and TGF-beta. IL-6 is sort of that pro-response, pro-inflammation, pro-attack of a pathogen, whereas TGF-beta, again, is one of those homeostatic return to baseline cytokines. And you can see, if we look now at day 2, that the IL-6, there's a large induction, but then almost immediately gets dampened down, while there's this gradual increase in TGF-beta over the 14 days that we looked here. We looked at signaling. So immune cells will have these signal transduction pathways that alter their effector function, turn them on or off, change their response to an infection. And this WNT pathway is an immune pathway. And at day 4, you can see a strong induction. You've got, we measured the fold change increases of all these proteins up and down this pathway from the cell surface down to the nucleus and altering its function. But at day 7, 10, and 14, all these dashed lines indicate a lack of response between an infected bird and a control when you compare those two. So there's no real induction of an immune response after that day 4. So we've done a lot of work in the lab here at ARS and in my lab at Delaware previous to that. And we've sort of traced this tolerogenic response that we call it to salmonella in chickens. And we've broken it down into these 3 stages. So you have stage 1, where you have, it's 1 to 2 days post-infection, where you see what you would normally see in a human, where the immune system recognizes salmonella as something that's out of the ordinary, that's foreign. and they mount a classic immune response, pro-inflammatory. These immune cells come to the site of infection. You've got this signaling that's happening in the immune system, telling the system to activate. You see metabolic changes, which are representative of an immune response. And then 3 to 4 days post-infection, you start to see a shift. So you've got those homeostatic dampening cytokines. Those regulatory cells start to take over. That signaling that would modulate an immune response starts to happen. And you have this, what we call tolerance or anti-inflammatory effect. And then 5, 7, 14 days out, the system is back in balance. So you have T regulatory cells, interleukin 10, a maintenance signaling, a neutral metabolic response, and the system's not inflammatory, anti-inflammatory. It's basically returned to baseline. So what the chicken are doing is that they're seeing it, they're actively generating a tolerance, and then they're returning to homeostasis. Not something we would see in a human system where we would see a strong inflammatory response, a strong innate and adaptive response. You'd get that active attempt to clear the pathogen through immune responses, through diarrhea, through things like that, to try to clear the pathogen. Not something we see in the chickens. And other people have studied this phenomenon as well from different perspectives. This data here is from Huaxian Zhao, the University of California, Davis. And he looked at the microbiome of broiler chickens that were inoculated with salmonella. And here is he's measuring the colony forming units per gram in the cecum, those blind sacs there off the large intestine where salmonella like to take up residence. And he found very consistent numbers of salmonella in the ceca from day 3 post-inoculation to day 21. And then he did the standard microbiome type analysis of richness, diversity. Saw a little bit of actually increase in richness in the microbiome with the salmonella infected birds. No difference in diversity of the microbiome. And when he plotted out the microbiome profile of salmonella infected and not infected birds, they actually scattered all over the plot, which means you couldn't distinguish from the plot, from their microbiome profile, which birds had the salmonella and which birds didn't. So basically, the salmonella are establishing themselves as part of the commensal microflora of the bird. So and what we saw from the immune system side was that was an active transfer from inflammation to tolerance to homeostasis, where the salmonella survived. And then they didn't have to worry about the immune system anymore. And here, looking from the microbiome perspective, there was no major shifts in the microbiome itself, but the salmonella were able to take up residence and persist at least through 21 days without major disruptions of the microbiome system as a whole. So what the salmonella seems to be doing is that they're inducing an immune response, but enough for them to establish themselves. Salmonella take advantage of inflammation to develop an ecological niche in the gut. So there's evidence that they actually use the immune system to knock out some of the microflora, use those bugs that are getting taken out as a food source, and then setting up a niche for themselves there. And then they're able to persist over the long haul. And persistence is really the name of the game. This study by Tim Johnson, he's a professor at the University of Minnesota, really good microbiome genome scientist. And what they did in this study was they looked at the persistence of a salmonella vaccine against Salmonella typhimurium through the food chain, basically from birds to product to humans. Actually, they went all the way up. So they looked at FSIS samples at the plant, FDA samples of product, and then they actually looked at some of the CDC sequences. So they got the sequences from sequences that were put online, and they had markers of the vaccine strains that were used. And they looked over a number of years, number of samples from chickens and turkeys here, and they saw the number of chicken source isolates of vaccine origin, a number of turkey source isolates of vaccine origin, and they found in total 6.6% of the isolates from FSIS samples were actually of vaccine origin, and 40% were of vaccine origin from turkeys. And then when they went to the FDA sampling of the products, they found 8.3% of the samples they looked at were actually vaccine origin. So these are live attenuated vaccines that are used to reduce the salmonella loads in the live birds. And the goal is that you'd reduce those serovirus specific loads. You'd reduce the amount of salmonella that goes to the plant, ends up in the food, ends up potentially causing people to get sick. But this bug is so persistent that it actually persists and is detectable when we give it to the birds as a vaccine. And they actually found that 0.1% of the CDC generated sequences from salmonellosis cases could be traced back to a vaccine strain of salmonella. So extremely persistent bug in chickens. So when we're thinking pre-harvest food safety, which is what my unit does, we're thinking, how do we eliminate these foodborne pathogens in the live animal before they go to slaughter? And we have to think about that in the context now of what I've described to you of a salmonella being a commensal, establishing tolerance with the immune system and establishing an ecological niche in the microbiome. So if we were to create an analogy in humans, it's like saying to a person, we really don't want you to have this streptococcus or this lactobacillus in your gut anymore. It doesn't matter. We know it doesn't affect you. We know it has nothing to do with your health, but we don't want you to have it in there. So get rid of it. Well, how would you go about doing that? How would you identify it? How would you modulate the immune system? How would you vaccinate against it? Maybe nutritional, maybe antibiotic, but we can't use antibiotics, maybe antibiotic alternatives. The amount of basic biology we would need to understand of how commensal microflora establish themselves, what timing of exposure, how do vaccines work, how do we break tolerance? There's a lot of basic biology that goes into an attempting something like what you see on the screen there. So that's really the challenge, a unique challenge that we face with chickens, especially with broilers. With cattle, there's a similar phenomenon going on. Cattle respond and swine respond a bit more to salmonella than chickens do, but salmonella are an intracellular pathogen, and they can survive inside of cells, and sometimes they're able to subvert the immune system enough to survive inside some of those phagocytic immune cells. So the other thing that cattle have is they have a lymphatic system. So those immune cells will take up the salmonella and they'll go to lymph nodes. And lymph nodes are a problem when it comes to contamination during processing, where we can't always get all the lymph nodes and all those concentrated sites of foodborne illness out of those carcasses before we generate the ground beef and things like that. So you might be asking, okay, so if this is such a difficult problem and we're having such a tough time dealing with salmonella, even though the poultry industry has reduced salmonella loads by 50%, it's estimated over the past few years, why are there other countries, like why is some of the European countries saying that they're salmonella free or they treat salmonella as an adulterant and they're able to eliminate it? And there's a number of reasons for that. One of the reasons is just the scale of the issue. In the United States, on average, at any given time, we've got about 500 million broiler chickens being produced. Some of the countries like Denmark, they're looking at maybe 20 million, Finland, 14 million. So they're really operating at a totally different scale than we are. Sweden's another example, operating at about this level. And there's been some really good work, including here example of a report from the University of Georgia Cooperative Extension Service about the differences between European production and testing and intervention compared to the United States. So there are clearly production differences of scale and type. There are processing differences, and that's a big one. There's also sampling differences. So in the U.S., we'll do things like whole carcass washes in Peptone water. In Europe, they'll do neck skin sampling, or sometimes for export, they'll actually take a whole chicken breast, flame it, and then go inside of the muscle to take a sample to test for contamination. Whereas you're kind of set up in that type of technique to not find a positive. So there's quite a few differences all up and down the chain when it comes to Europe and the United States. So we're not really comparing apples to apples when we compare how the production is done and how the testing is done. And really where the rubber hits the road for this would be reduction in the number of human cases. But the last good data that I could find was from 2022. And when it comes to salmonellosis cases, we're very much in the same boat, Europe and the United States, 16.3 cases per 100,000 in the US and 15.5 cases per 100,000 in the EU. And the other thing to think about with all of the strategies and restrictions and interventions and regulations that go into foodborne safety is, what does that do when we're talking about this is a food product and we're attempting to feed the world? And food prices saw this huge run up during 2021, supply chain disruptions, all kinds of issues with getting animals to processing, getting the food to the consumer. And luckily, the inflation has come down quite a bit when it comes to commodity prices. But in the European Union, they're still having this issue with food prices more so than other places in the world. And despite the fact that the US is, we spend the least as a percentage of our disposable income on food and compared to the rest of the world, people are still very price sensitive in the United States. You can see this graph of the ever increasing consumption of chicken, whereas beef has gone down and pork has sort of maintained its level. And really, this is, in my opinion, is an indication of price sensitivity. Chicken productivity, chicken prices have maintained their affordability. Beef has gotten quite expensive, especially now. And the US consumer is price sensitive, and there are people in this country that are food insecure, and any major shift in food costs can have a profound impact. And I think that's always something to keep when we're talking about a One Health perspective. And part of that One Health is humans and human nutrition and feeding humans, as well as doing it safely and producing animals in a humane and safe way, is that we think about the cost and how some of our interventions might affect affordability. So I'll leave it there. Thanks for the opportunity to speak today.
Thank you so much, Dr. Arsenault. So there is a Q&A function at the top of the screen, and I'll give it a couple of minutes here. If anyone has any questions, please feel free to drop in the chat. While people are thinking about questions, I do kind of have a general kind of question regarding kind of forecasting and seeing that, as you mentioned, the idea of removing something that can be viewed as a commensal in these livestock species. Are there any kind of new technologies or innovation areas that ARS or others are potentially look at to try and kind of gather that specific kind of targeting aspect of these bacteria?
There's a lot, and if we knew which one was going to be silver bullet, we'd focus on that one. But there's a lot that's being tried. We're bringing back the old technology that used to be in vogue. That's now back, I think, in vogue, like the bacterial phages. A lot of work on antibiotic alternatives, botanicals, phytogenics, vaccines, different strategies to attenuate vaccines or to have killed vaccines. Yeah, it's, in my opinion, what we need to do is, like I said, about feeding the world, is we certainly can't go back and reduce productivity. And my research, we've done a lot with antibiotic alternatives, and I think we can maybe modulate and feed back in some of the responses so that we can make the birds a little bit more responsive and maybe lock in that early response where they actually do respond and start to react to the salmonella, and try to avoid that tolerance from taking place. So that's the kind of approach that we're looking at, is okay, you're obviously recognizing it, keep doing that, keep carrying on, rather than tolerate, and then we're in that difficult task of trying to break the tolerance, which is a difficult task.
Great, thank you. All right, I'll give it, I don't see anything in the chat as of now, so I'll give it just another minute here. And if there aren't any questions, then we can wrap up things a bit early. I will just note that the next quarterly meeting will likely be December, January time. So please keep an eye out for updates, probably coming more towards November for the next quarterly meeting. And if I've still not seen anything in the chat, so I'll give it one more minute, and otherwise I appreciate everyone for attending, and we'll see you at the next quarterly meeting. All right, great. We'll wrap it up there. Thanks, everyone.